here we are dealing with a complex disease, multifactorial disease, a lot of data, a lot of variables. How to put order on that? What is the role of bioinformatics, computational biology from your perspective in order to help to this complexity?
You know, Pablo, the bioinformatics play a crucial role in this aspect because it gives the opportunity to integrate data and to analyze the data within the layers and across the layers to generate profiles either for the disease or for the patient. So we can bring together MRI imaging data, genomic data, other omic data as well, and lifestyle data to generate the so-called profile. And this profile, we hope and we believe that it is a stepping stone, a very good stepping stone for AI tools for prediction and risk monitoring.
Okay, but here we have a special situation. We have people with MS, and then we have what we believe is our enemy or not, which is a virus, which is interacting inside of the body, meaning how you manage interrogating the human body, a virus that is interacting and all the war inside of the body.
It seems very challenging for us because up to now we were working with network analytics, but now it seems that we should work with networks of networks. Mm-hmm. So we try to see how the networks around the proteins of the EBV virus, they interact with the host proteins. So we have like a war between the networks, interaction, and we try to find the routes, the shortest paths, as we say in network science, in order to pass from the EBV proteins at the end to the MS-related proteins. And this network of networks interaction is very challenging also from graph theory and other mathematical fields in order to give us hints and next to these hints, we will give them to the experimentalists to, let's say, make them focus better.
Okay, maybe we have the wrong idea that we have, the human being, meaning a single body, and then the microorganism. But it's not truly the situation because, our body is plenty of bacteria. We have many viruses living with us. Our genome is composed 1/3 from viruses, meaning it's not so much clear the difference between our DNA and microorganism DNA. We are living together in symbiosis, sometimes but sometimes degenerates in diseases. Meaning, do you think that this approach of bioinformatics and computational biology can help us to clarify what is good, what is bad, why things happen, in this specific case in MS and relationship with the Epstein-Barr virus?
It's great that you are noticing this because these days I was thinking that taking into account that EBV is prevalent, highly prevalent in humans, we can talk perhaps like a new normality. Mm-hmm. So as you said, we live together, and this generates a new situation. We think that we can identify interesting findings. I don't know, I'm not sure if at the end of the day we will be able to solve the mystery, but I am sure that we will contribute to its solution because we are able to analyze omics to find signatures, disease signature, virus signature, the symbiosis signature, and also we can find other signatures from other viruses, as you say. Some viruses, they are good, some other, they are not. And sometimes we can use one virus to help get rid of the other one. Mm-hmm. And all this systems analysis, we call it, from our side, systems bioinformatics. So we don't want to work only on the classical side of the story, on the bioinformatics, but we like to see the relationships as markers, not only the entities. You know, the classic thing is to find genes, proteins, highly expressed or underexpressed. But when you have networks, you see that as the progression of the disease goes, you see that the entities, the genes or the proteins, change friends. So this change is a marker. And we have the opportunity to see the so-called network rewiring, how network changes throughout the progression of the disease. And we believe that these relationships could complement the other markers that we can have, and also we can have relationships as markers. But I have seen also very interesting works from your lab. And it's great to see that you're working also with this multi-source and multi-layer networks. And I believe that we have many in common with your work. I liked very much the Boolean logic that you developed. Would you like to elaborate a little bit on this?
Oh, of course. You know, networks are a very nice way to summarize very complicated information. You know, I think that everybody has personal experience on network because we have social networks, not only in the internet, but your friendships, your acquaintances. Meaning the network can be composed by genes, proteins, cells, organs, people, you know, and the relationship are the interactions, meaning we have the nodes and we have the edges, which we define this interaction based on statistical methods or any other kind of metrics. A further step is when you relate networks that are different but you put together. For example, trying to address different biological scales, from genes to proteins, to cells, to tissues, to the whole body. Okay, there are ways to relate one to the other. It's amazing that just by applying these, let's say, classical statistical methods for networks, the data from each of the layers clumped together. Okay, you can see nicely in the statistical analysis, and then you relate one to the other, and after that you study how the information flows from one layer to the other, because otherwise it's difficult to relate what's happening in the genes, what's happening in the cell and in the tissue. And then networks may look like statics, meaning just a picture, but you also can analyze along the time, you know, doing a dynamic analysis. In this case, we apply these basic rules of logic Boolean networks, which is a way just to operate and model how the networks evolve over time. And this is very important, exactly for what you have said, because most likely the genes of the cell are not changing so much. What they are changing are their friends, the relationship they establish, meaning how networks evolve over time is what maybe is the most important thing, more than that single gene, a single protein. Don't you agree?
It is exciting that we have this common sense on the analysis of the networks. And it's very nice because we see that the work packages that we are working, your work in the work package 6, and we're working in the work package 9 of the EBV MS, they are very complementary. So we feel that we can give to your work package the so-called integrated profiles for the AI systems that you will develop, and also some network views that you can further analyze. And also we can take for further analysis your multi-layer networks to try to find candidate repurposed drugs. You know, candidate repurposed drugs is part of our research in our department. We try to utilize these signatures, omic signatures or knowledge-based signatures, in order to find candidate repurposed drugs. And we say candidates because it is a computational analysis. So we try to highlight and shortlist good compounds, but these compounds, the short list of the compounds, they need to be experimentally validated. But it is very, very nice, and sometimes it is exciting for us when from, let's say, 100 candidate compounds, we shortlist in 5. And these 5, they work the bench, and they are ready to go for a preclinical study sometimes. So we feel that we can contribute also to this drug discovery part of the consortium, and we have a very nice complementarity between the two work packages. So have you worked in this direction also? Do you find that the findings that you can have from your work package, they can give a boost also in the direction of drug discovery?
Yeah, exactly. Meaning this is more than just a toy to use and understand the disease, but we can use for improving diagnostic and therapeutics, and they will benefit people with MS. This is at the end what is important here. Yes, definitely this can be used for drug discovery in order to understand this relationship between the different components, the gene, the protein, the cells, and try to identify what is first, which one are related, how this evolve, and by this way try to find different strategies to stop down this cascade that ends up in inflammation in multiple sclerosis. Now what is fascinating is when we confront the gene network from the immune system, healthy people, or people with MS, with the virus, we are seeing a lot of commonalities. And one is interacting to the other eventually for good, but in the case of MS, most likely for bad. And we believe that these interactions are the points in which we need to find therapeutics in order to reestablish the network or evolve the network to a situation in which is not promoting inflammation anymore. And for the reason, these interactions inside of the network are becoming candidates. We can analyze with statistical methods and also with artificial intelligence, because networks are complex, and in order to define which is the best protein to target with our therapeutics, it's important to understand very well the role, the position in the network, and how to modulate in order to stop inflammation. But then also, do you think that we can use for diagnosis?
Yes, for sure, because we know we are looking for signatures, and these signatures can be complex patterns of biomarkers, so they can be used for diagnosis and give more evidence, information in the diagnostic part. When we are talking about networks and attacks on networks, everybody's mind goes to computer networks. But there are also attacks in the biological networks. So when there is an attack in a biological network, could be a mutation or a change in the gene or the protein, as you said. But there is also the attack in the disease network. And this is the drug that we are giving. So sometimes we are attacking, or we try to attack to a disease network, trying to reverse. And of course, we can have these signatures to monitor the response of the drug or the success that we can have, etc. So it is both sides that we can contribute, the diagnostic, monitoring of the drug treatment response, and of course, the highlighting of candidate drugs.
Yeah, most of the time we have in mind, or our goal is to come back to the healthy state, meaning come back to the past. But biology, this is difficult because time is against us. And for the reason, some of the strategies that we need to envision is how to evolve the network that start in a healthy state and move to a disease state, which is MS, to another state in which maybe it's not 100% healthy, but it's not creating so much damage. Yes. Meaning, because most likely coming back is very difficult.
Almost impossible.
Almost impossible. Time is time, age is age. And for the reason, we need to envision how to evolve the MS network to a state in which it's not producing the damage, and disease more or less is controlled. How do you envision that?
Yes, we have also in physics things like this. So we try to go, let's say, from a healthy state to another healthy state by passing the disease states. And of course, we can have also a scoring of what is healthy each time. And we can, of course, compromise sometimes. But it is very nice to see how we can go from healthy state 1 to healthy state 2, let's say. But sometimes, I don't know if you face also this problem, working with the networks, one tricky question is if we work on the correct network. Sometimes, there are easy to go networks, and some other networks, they are not so easy. So one question that we always have is whether we are working on the correct type, because we have protein-protein interaction networks, we have omics-based networks, we can have genetics network. So how you deal with this question each time when you have the opportunity to select many types of networks for the same, let's say, question?
Yeah, this is one of my main concerns, meaning data. The problem in biomedicine is that having data is difficult. High quality, quantitative data is difficult, but the most important thing, relevant data. Most of the data, when you check, is highly correlated between themselves, meaning it's not adding so much. Meaning you check several omics and all of them are highly correlated, but they are not adding information. And maybe the important things related with the interaction between the Epstein-Barr virus and MS, we are unable to measure at this time. Meaning this is my main concern that we have at this time. Okay, there are some approaches here, ways. We have the data-driving approach, meaning you do everything and you apply this bioinformatics network analysis and you see what pop up from this analysis. Or you have hypothesis driving, meaning, okay, there is a lot of information and knowledge has been created in the interaction between herpes virus and the immune system. And okay, we can drive in order to ensure that we are collecting the right information. And for the reason, technology here is very important. Being able to interrogate the immune system in the right place, meaning we can perform genetic studies and immunological studies easily in blood, but interrogating what's happening with the immune response inside of the brain, this is truly a challenge. New technologies are being developed time by time, are becoming very powerful. For example, right now, is very easy, or even almost mandatory, to interrogate the whole transcriptome, meaning the RNAs for the cells, RNA-seq, and you do one in a shot, all of them. Now proteomics is moving to this scale. Then we are going to have metabolomics, lipidomics, everything, and also the ability to collect this information or associate this information with specific cell subtypes, meaning always biology is highly specific. Little changes I surprised, for example, in chemistry. In chemistry, you just change the one atom, meaning from hydrogen to a methyl group. The molecule behave completely different, even if you change the dexterity. Of course, if this happens at the atomic level, of course, at the molecular level is even more important, meaning we need to be precise. In quantification, precise in the space, meaning whether we are measuring blood, whether we are measuring from the lymph node, whether we are measuring from inside of the brain, and be highly precise and quantitative. With that, I'm sure that we're going to contribute with some important discoveries.
It's fantastic, and I'm sure that with these networks that we will both develop, we will give explainability to the AI models that will be developed. So it is one thing to have a black box saying something, it's another to understand what is inside. And I think that in some cases, these network approaches, they will give explainability to the models, and then they can be adopted from the other scientists as well, because this is also important. Don't you think so?
Yeah, what you are proposing is extremely exciting, because AI is very powerful, but we don't know how this works. Meaning we know the technology, but not the explanation they are providing to us. Make AI explainable is not so easy at all. And what you're suggesting is perfectly fine, meaning you do a discovery with AI, which is amazing, but you don't understand, and then you explain how this happened with bioinformatics and network analysis. By this way, of course, we can make big progresses and help to discover new therapeutics.
I'm looking forward to this wonderful collaboration, Pablo.
Okay, it was a pleasure. Thank you, George.
It was a pleasure also for me. Thank you.